Good morning, everyone. We welcome you once again to this new Conda Lab talk. So today we will talk about an important topic in food safety. I will introduce myself. My name is Aletia Cruz, and I am one of the microbiology product managers at Conda Lab. And for those who are joining us just for the first time, we just want to let you know that we have started, more than 1 year ago with these kind of lab talks that are webinars we prepare bi-monthly to discuss, selected topics of microbiological interest. So today it will be the turn of a great unknown but important bacteria that causes foodborne infections. So surely by now, most of you will be familiar with this platform, but just as a quick reminder, that GoToWebinar has a chat box for you to send us your questions or doubts throughout the presentation that we will address at the end. So today we will talk about Yersinia and the disease it causes, the current regulatory environment in the European Union. Then we will go on to review the ISO method for the detection of these bacteria. And just to close, we will discuss current and future challenges in the industry regarding these microorganisms. So, finally, as I mentioned, we will end with the Q&A session. So I think we should start by getting to know the bacteria and the disease. So we can go back all the way to the 1880s, which is when the story of Yersinia began, when a strange disease that affected guinea pigs was reported and that evolved to what we know today as, Yersinia. So given it's a member of the Enterobacteriaceae family, Yersinia is a gram-negative bacteria, oxidase negative. They are non-spore forming bacilli and facultative anaerobes that ferment glucose. All of this information is important for its survival, as we will see later on. And a curious fact is that because Yersinia is smaller than other Enterobacteriaceae, when these colonies grow at around 37 degrees Celsius, they often appear in smears like cocoa bacilli. So today, we know that the genus Yersinia comprises 13 species, of which 4 are pathogens. Yersinia pestis, which was the causing agent of bubonic and pneumonic plagues. Also Yersinia rukeri, the cause of disease in salmonids and other fish species that are not relevant for humans. Yersinia pseudotuberculosis, which is a rodent pathogen that can cause infections in humans, and of course Yersinia enterocolytica, a versatile intestinal pathogen that is today's main focus. So Yersinia pestis is transmitted through bites or aerosols, whereas Yersinia pseudotuberculosis and Yersinia enterocolytica are foodborne pathogens. And while Yersinia pseudotuberculosis was once quite common in Europe, today it is rare. And conversely, when Yersinia enterocolytica first emerged as a human pathogen, it was considered rare, yet it is currently the most prevalent Yersinia species among humans. And for this reason, we have focused this webinar on the latter. So, specifically referring to Yersinia enterocolytica, the optimum growth temperature is approximately from 28 to 30 degrees Celsius. At this temperature, it can double in half an hour, and if the temperature drops, the time lengthens. But the growth still continues. For example, at 1 degree Celsius, it would take almost 2 days to double. So this is a serious problem because it tells us that Yersinia enterocolytica has been able to adapt to cold temperatures, and therefore the bacteria are psychrophilic, which means they can resist refrigeration and freezing temperatures, as well as repeated freeze and thaw cycles. So, of course, this is a reason for concern to the industry, since Yersinia enterocolytica also persists longer in processed foods rather than raw, possibly because of the availability of nutrients, given Yersinia is not a very demanding bacteria, and maybe also by the presence of other extremophilic bacteria that could somehow restrict the growth of Yersinia. So about the pH that we will talk about later, but just to mention, the optimum is 7.6. Yersinia enterocolytica is also a problem in the health sector, specifically in blood transfusions due to its ability to proliferate and release endotoxins, which is expected because it's a gram-negative, of course, in these blood products stored in refrigeration, and all this without altering the appearance of the products, which is very worrying. So Yersinia enterocolytica is heterogeneous and therefore has been able to be divided into subgroups according to its biochemical activity and the lipopolysaccharide O antigens, as we can see here on the table on the left. The vast majority of isolated pathogenic strains correspond to biotypes 1b, 2, 3, 4, and 5. These have a different geographical distribution. As we can see, biotypes 1b08 have been found mainly in North America, while 403 predominates in Europe, and in fact has been confirmed as the strain in the last outbreak in Sweden, on which we will comment later. So in these main pathogenic strains, 1b is the most virulent biotype, followed by 2 to 5 with an intermediate virulence, if you will, and biotype 1a, which is the least virulent and mostly considered non-pathogenic, although some strains of this biotype have been related to patients with gastroenteritis. So therefore, this could mean that they would act as opportunistic pathogens. So enteropathogenic Yersinia infection has a common manifestation with other foodborne infections, the most common one being gastroenteritis. However, on some occasions, it can cause symptoms very similar to appendicitis, thus leading to a wrong diagnosis. Less common complications are autoimmune sequelae, pneumonia, and bacteremia, which is when bacteria enters the bloodstream, and it is very dangerous since Yersinia bacteremia has a fatality rate between 30 and 60%. So Yersinia enterocolytica enters the intestinal tract through the ingestion of contaminated or food or water. The exact infectious dose is unknown, but it could exceed 100 CFU. The first body defense seems to be gastric acid, since it's been observed that in people with gastric hypoacidity, which, as the name suggests, is a decrease in acidity of the gastric juices, the infectious dose may be lower. This incubation period is generally less than 10 days, from 3 to 7. The highest percentage of symptomatic infections occurs in children, especially those under 5 years, and the disease can last from days to 3 weeks, although chronic diseases such as enterocolitis can develop, and these could persist for months. So according to the ECDC, Yersiniosis does not follow a clear seasonal pattern. In this first graph on the left, that indicates the number of cases and the months in which they were reported between 2015 and 2018. We can, of course, see some peaks here, but these don't correspond with the months, with the highest number of cases in 2019, which would be this green line. So here we have highest from 2015 to 2018, and this would be the lowest month in 2019. So relying on information from the latest epidemiological report by the ECDC published earlier this year, we know that in 2019, yersiniosis was the fourth foodborne zoonotic disease most frequently reported in the European Union. So in this bar graph, we have the notifications of confirmed cases of zoonotic diseases. We can see, of course, the always trendy CAMP, Lobacteriosis, and Salmonellosis, but it is, for our purpose today, more interesting when we focus on these other infections. So let's take a look. Okay, so when we focus here, we see that there really isn't a huge difference between staph infections and Yersinia, yet staph is in the public eye. We even have Listeria more in mind when it's only a third of Yersinia cases. And as we saw previously, there are two pathogenic Yersinia species for humans. However, Yersinia enterocoletica was the cause of almost all infections. And just to comment quickly about recent outbreaks, in February 2019, there were investigations of an outbreak that began in Sweden, but also affected Denmark. And the same strain of Yersinia enterocoletica was detected and confirmed by genomic analysis. It was the biotype 403. And fresh spinach was pointed as the source of the outbreak. So later in May of that same year, another investigation was launched from Norway, and again, Yersinia enterocoletica serotype 03 was found. Sweden reported cases with isolates that were genetically close, and this was an outbreak that could have had the potential to involve several countries. However, it is not known if there was any exposure in common between the two countries. There are currently actively ongoing investigations. In South Korea, they have detected Yersinia enterocoletica in kimchi, that's a fermented product, vegetable product from China. And on the other hand, in Norway, there is an increase in yersiniosis since the end of April. And based on the geographical area of those infected, it seems that it would be a widely distributed product. The culprit again is Yersinia enterocoletica, surprise, surprise, serotype 03. So now we have very clear that yersiniosis is a disease that is transmitted between animals and humans, mostly through food. So products that can harbor Yersinia enterocoletica include meat, whether it's pork, beef, poultry origin, dairy products, and vegetables. The latest outbreaks being reported due to the consumption of this type of fresh produce. And well, Yersinia is also found in a wide variety of ecosystems such as soil, vegetation, lakes, rivers, wells, where it can persist for long periods of time. The vast majority of isolates obtained from these environments or the environment lack virulence markers, so their importance in human or even animal health is uncertain. Asymptomatic pigs are the main carriers of Yersinia enterocoletica and thus is frequently isolated in slaughterhouses, being the O3 serotype, the most common, that has been found mostly in the tonsils, intestines, lymph nodes, and feces of these animals. Other sources, of course, would be farm animals, domestic pets, and wild and captive animals. There are intrinsic and extrinsic factors that contribute to the survival and growth of Yersinia in food and that have a direct consequence in promoting outbreaks. The more important intrinsic factors include pH, nutrition, and water activity. So Yersinia is capable of growing in a wide pH range between 4 to 10. We have already seen that the optimum is 7.6. And the vast majority of foods have an acidic pH. So the tolerance of these microorganisms to higher pH values is of little importance for its survival. However, at very low pHs, its growth can be slowed down or even stopped, inhibited, depending on the acid. Yersinia is not a demanding organism when it comes to nutrition, so that's another plus for Yersinia. And finally, the tolerance to salt is another factor that depends on the concentration and storage temperature. In commonly levels we find in food, the salt by itself would not prevent growth. So other barriers are required. Not even nitrates in certain concentrations by themselves can inhibit growth of Yersinia enterocolytica. So on the other hand, the main extrinsic factors are the temperature and the gas atmosphere. We know that Yersinia is psychrophilic and can grow at low temperatures even at a slow rate. And on the contrary, Yersinia is sensitive to heat. Its maximum growth temperature is between 42 to 44 degrees Celsius. So usual heat treatments in food processing destroys it without difficulties. For example, pasteurization at 72 degrees Celsius for 15 to 20 seconds destroys it with no problem. And also by being a facultative anaerobic, Yersinia can grow in modified atmospheres, for example, vacuum packaged foods. Although their growth slows down, it's still there. And when it comes to the environment, Yersinia enterocolytica is susceptible to chlorine in general, although there are, of course, some wild strains that have shown some type of resistance. So we will quickly review now the regulatory environment for Yersiniosis in the European Union and the European Economic Area based on data from the latest ECDC epidemiological report published this January. So Yersiniosis is contemplated within zoonoses to surveil and are included in Annex 1 of Directive 2003/99 EC on the monitoring of zoonoses and bacterial agents. And at a national level in Spain, Royal Decree 1940/2004 of September 27 also includes Yersiniosis and its causative agents within the bacterial zoonosis to be monitored. Just to go a bit outside of the European Union, in the US, the American CDC monitors Yersinia enterocolytica through its active surveillance network for foodborne diseases called FoodNet. And obviously, each country must indicate how and if it carries out the surveillance according to its own epidemiological situation. According to the report of the ECDC in 2019, the data for Yersiniosis was reported from 29 countries. Among these, it should be noted that notification is mandatory in 23 of them, while in 6 member states, the notification is voluntary, or its notification was based on another type of system, such as was the case of the UK at that pre-Brexit time. A curious fact is that in the Netherlands, there is no surveillance for Yersiniosis. As you can see here on the map, there is no data available. In addition, in 3 countries, which are these 3 in gray, Spain, France, and Italy, it is not possible to estimate the percentage of notifications because the surveillance systems are not unique throughout the territory. When specifically referring to Spain, it is because it does not send data from all of its regions. So the lack of mandatory surveillance and national coverage in a quarter of the member states, of course, contributes to low reporting rates in these countries. And it would be logical to think that due to all of these circumstances, the number of reported cases is underestimated. And despite it being the fourth disease with the highest notification, it can be concluded that the most of the member states actually carry out a passive surveillance for Yersinia instead of an active one. So continuing with this report, more than 7,000 confirmed cases of yersiniosis were reported in 2019, being almost all infections caused by Yersinia enterocolitica. Luckily, the mortality rate is quite low. There were 2 deaths, both men over 65 years. Following the trend of previous years, Germany and France were the ones that registered the highest number of cases, representing between them almost half of the total number of cases that year. And for its part, Finland had the highest rate per 100,000 population, followed by Lithuania and the Czech Republic. We once again confirm, as you can see in this graph on the right, that the age group mostly affected by Yersinia infection were infants and children up to 4 years of age, who accounted for almost a quarter of all confirmed cases. Outside of the European Union, the American CDC estimates that in the US, Yersinia enterocolitica is the cause of about more than 100,000 infections annually, 640 hospitalizations, and 35 deaths. And again, their data reinforces the conclusions of the European Union that children are the most frequently affected. And they did found seasonality of yersiniosis being in winter, where there are some spikes in infections. So now we will go on to review the standard method for the detection of Yersinia enterocolitica. As usual, in the food industry, culture methods continue to be the golden standards for pathogen analysis. So this method may seem a bit confusing, but let me tell you, it is really just to use the same media repeatedly to try to find a strain of Yersinia enterocolitica. You will see what I mean by this. We begin, of course, with the test portion of, for the analysis and the initial suspension. PSB broth is used as the diluent for the preparation of this initial suspension, and the media must be preheated to room temperature before use. You have to mix 25 grams or milliliters of the sample with 225 milliliters of PSB broth and homogenize for 1 minute. With this suspension, direct plating can be carried out on a selective agar, which in this case would be the syn agar. The plates incubated at 30 degrees Celsius for a day, and the number of syn agar plates would depend on the level of expected basal microbiota in the samples. But if we want to continue with the enrichment, possibly because we are not sure if we have a large quantity of Yersinia enterocolitica or not, we have to prepare another suspension, this one in ITC broth. So again, it's just mixing test portion with the ITC broth. Now we move on to the enrichment step with both suspensions, one in PSB and ITC broth. These must be incubated at the same time at 25 degrees Celsius for what would be 2 days, 44 hours. This would be the enrichment step. So we have our enrichment samples, and once the incubation is complete, a portion of the product enriched in PSB broth is transferred onto a potassium hydroxide solution, and it is strict on syn agar. We repeat the procedure, of course, with the product enriched in ITC broth. It is crucial for the correct operation of the method that the potassium hydroxide is prepared the day before its use. So the plates must be placed in an inverted position and incubated in a maintained temperature of 30 degrees Celsius for a day. After this incubation, the syn agar plates are examined for the presence of characteristic colonies of Yersinia enterocolytica, which should be done with the help of a stereo microscope equipped with dark field illumination. On syn agar, pathogenic Yersinia enterocolytica appears in the form of small-sized colonies around 1 millimeter or smaller that are circular and smooth in appearance and have a central zone with a well-defined dark red border, usually known as bullseye. The surrounding border of the bullseye should be translucent and, when examined with obliquely transmitted light, non-iridescent. For confirmation, Yersinia strains are required as positive and negative controls to help distinguish between pathogenic Yersinia enterocolytica from other Yersinia species on syn agar. For the confirmation, the ISO calls to select 5 colonies considered typical of pathogenic Yersinia enterocolytica from each syn agar plate, and strict this again on syn agar under conditions that allow the development of well-isolated colonies. So now we have strict 2 times on syn agar. The control strains of Yersinia enterocolytica of biotypes 403, 209, and also biotype 1A and other Yersinia species are also strict to compare the morphology of the colonies. This is incubated at 30 degrees Celsius for a day and then examined for the presence of characteristic colonies, again using a stereo microscope, and the morphology of the suspicious colonies is compared with the control colonies to achieve a better distinction between typical and atypical colonies. And if we observe mixed cultures with the presence of typical colonies, these have to be again subcultured on syn agar, would be the 3rd time, and incubated under the same conditions we have discussed before. A pure culture representative of the initial typical colonies is needed, so the selected ones on the syn agar would be now strict on non-selective agar, such as nutrient agar or blood agar or even TSA, incubated at 30 degrees Celsius for up to a day or more until the observed growth is satisfactory. These pure cultures are used for both the determination of pathogenic species and the biochemical confirmation. For the determination of pathogenic species of Yersinia, we have assays for the detection of urease, hydrolysis of esculin, detection of the PYB virulence plasmid, and the detection of pyranimidase. A strain corresponding to Yersinia is considered pathogenic if it is positive for urease, negative for esculin, and pyrazinamidase. Also, the presence of the PYB virulence plasmid is a strong indication of pathogenicity. After determining whether the species are pathogenic, Yersinia enterocolytica is confirmed by the rest of the tests of lysine decarboxylase and arginine dehydrolase, phenylalanine deaminase, sucrose, sorbitol, rhamnose, and malleolose fermentation, carb fermentation, and the Simon citrate, which is optional. There are, of course, other assays included in the ISO for the full biological typing with assays for xylose, xylene, twin esterase, and trehalose, and indolose. Once we have the results of all of our tests, it is considered that we have detected pathogenic Yersinia enterocolytica if at least one colony is positive for urease, negative for esculin and pyrazinamidase, and presents the characteristics indicated in the table. So it is evident that in the isolation of Yersinia, syn agar is essential, since this media contains selective agents such as ceftazidime and novobiacin that improve the inhibition of normal enteric organisms, as well as irgazan, deoxycholate, and crystal violet that selectively inhibit gram-positive and negative bacteria. To picture how we would observe strains of Yersinia in syn agar, here we have an image where we have different species of Yersinia, both A, B, and F are Yersinia enterocolytica, but in all of them we can see the bullseye thanks to the fermentation of mannitol in the presence of neutral red, that is this red center with the well-defined edge, and then this outer edge being transparent and the appearance overall being, well, glassy. And this image clearly proves the reason to make the rest of the need to make the rest of confirmation assay tests, because as we see, Yersinia enterocolytica can be very similar to other species of Yersinia. There are, of course, several difficulties associated with the isolation of Yersinia food and environmental samples. Traditional methods that use culture media have limitations such as long incubation times, the ability of Yersinia enterocolytica to persist in samples in a viable but non-culturable state, which is an additional problem. It is a long and honestly tedious process, but it is necessary to be able to carry out an identification between species and discriminating between pathogenic and non-pathogenic strains, which is not possible only with a culture. So to conclude this webinar, we will comment a bit on the current situation of the industry and what at Conda we think we could or we might expect in the future regarding Yersinia. So the public health importance of Yersinia enterocolytica must be taken more seriously, since it is the main cause of yersiniosis and its detection in food is complicated and of great concern due to the fact that Yersinia is able to survive and even develop at refrigerated temperatures. For more than 2 decades, traditional methods have dominated its identification. However, this entails delays in obtaining the results. And of course, as a response, molecular methods such as ELISA, PCR, microarrays have been introduced. However, these methods, despite being faster and precise, its usefulness is limited given the genetic similarity between virulent and non-virulent strains, and since the markers used in the detection of pathogenic strains are also found in non-pathogenic strains, such as Yersinia fredericksensi and Yersinia christensenni. This is due to the extreme heterogeneity of Yersinia enterocolytica with other Yersinia species, and even with other members of the Enterobacteriaceae family, and the main problem for an accurate detection, of course. Therefore, this apparent low incidence of Yersinia enterocolytica in food may be due to the insensitivity of the available methods and thus added to the non-mandatory and passive surveillance that we see. We could even think of imitating a kind of the case of Campylobacter that shares with Yersinia a high incidence and a low lethality and mortality, but is now of a high relevance worldwide given the obligation to analyze CAMPy in chicken carcasses as a hygiene indicator. So we could establish Yersinia as a new indicator of hygiene and good practices, since many cases come from cross-contamination during the handling, processing, or even during the prolonged storage in refrigeration of not only meat, but also of vegetables and fresh produce that have been the culprits of these last outbreaks. So what is required in future approaches for the detection of Yersinia enterocolytica are not only new technologies, but rather than maybe the improvement of existing technologies for a correct monitoring of yersiniosis through a more precise and routine diagnosis of Yersinia enterocolytica in food. As usual at Condalab, we have available a complete catalog of products according to ISO in different dehydrated or ready-to-use formats for the analysis of Yersinia and Torcolytica. As manufacturers, we are able to control our products throughout the production process, from raw materials to the final product. We also test each batch to provide a consistent performance of our products. So now we will go quickly through the doubts and questions that you have been sending to us. So just a second. Okay, here we have. Are there other tests available for confirmation? Yes, there sure are. The ISO indicates that both the test for the determination of pathogenic species as well as the biochemical test can be performed by qPCR according to ISO 18865. 67, I'm sorry. But however, if a positive PCR result is obtained, then it is mandatory to proceed to the detection of P. racemamidase. So you have to go back a bit to this ISO. So, okay. Any recent outbreak of Yersinia pathogenic due to food products? Yes, as we have mentioned before, there are some ongoing investigations. We have one from South Korea, from kimchi, which is cabbage, fermented cabbage. And then we have another one here in Europe that it's in an ongoing investigation, so we still don't know the source of the product, but it is a widely distributed product. So, and maybe because of the outbreaks in 2019, maybe it is a fresh produce. Those ISO guidelines recommended testing of Yersinia in poultry and dairy industries for the release of the products. The ISO does not give any, this ISO 10273 does not give any guidelines for specific industries. However, for example, in dairy, if the product is pasteurized, you shouldn't be able to find any Yersinia because, as we've said before, Yersinia wouldn't be able to survive a pasteurization process. It is very sensible to high temperatures, but we would recommend to maybe validate your procedure to see if you have any contaminated products, and then you can put your own guidelines. In Saudi Arabia, poultry industry does not test for Yersinia. Yes, of course, because there are, as we've seen, other more, let me go back to the chart. There are other foodborne diseases such as salmonella and can be that are more worrying, also because of the mortality, lethality of the infections, the incidence that is very high. So of course, Yersinia is not going to be in the top pathogens to analyze. Okay, we have another one. Are there any alternative methods accredited for Yersinia? We have found quite a small supply actually in the market. There are, of course, some kits for the detection of Yersinia that aren't accredited, most likely due to the low monitoring and control of this pathogen. These kits can be quite expensive as well, and they are based on closed systems, so this restricts the regular use. So there's not a large offering for Yersinia, and therefore we must embrace the ISO method and all that it entails, the good and the bad. So we have some other questions, but we will address these via email. I don't want to take longer. Okay, so we hope that this Conda Lab was interesting for you, informative. If you require a certificate of participation, please request it through our email, micro@condalab.com. We want to invite you to join us in our next webinar, which will be after the summer. In the July edition of our newsletter, we will include the agenda for the second semester of the year. So there you can check out the next webinars, dates, and topics. Don't forget to sign up and also follow us on LinkedIn, where we also share this information. We hope you will have relaxing holidays and enjoy a lot. And from our side, well, that's it. We hope to see you in the upcoming Conda Lab talk. Until next time.